There's a couple of words that are a little uncomfortable for me in this and many of my colleagues who work in a developmental area, and maybe for many of you. We don't like to think about anything in development as permanent. But that word aside, it's a model, a way to think about the fetus adjusting to these exposures that has implications for future health. Here, he is very much focusing, as you can see, on organs largely associated with metabolic functioning, cardiac functioning. Sorry about that. But of course, the brain is also undergoing major development during this period and also adapting. And that's, of course, very relevant to ASD. So as we know, there's tremendous brain development in the prenatal period. And again, it's one of the reasons why this FOAD model is so useful for the ASD studies. So really briefly, I want to give a couple of slides just summarizing the phenomenal brain growth in this prenatal period. As we know, it's a time in utero of immense construction and vulnerability for brain development. There's the interplay of inherited genetic programs and in utero environmental exposures that determines the neuron's birth and death and cellular characteristics, and of course the formation and reformation of neurons and axons and dendrites and synapses. So just a schema of the phenomenal change in size and complexity of the brain during the in utero period. And then just 3 summary slides of what's really going on. The human brain contains 80 to 90 billion neurons. Most of those are produced by mid-pregnancy. And then neuronal migration, that is these neurons finding their rightful place so they can interact appropriately. Neuronal migration peaks around mid-pregnancy. And then finally, this is actually my favorite. The synaptogenesis that's going on, that's allowing, of course, a neuron to communicate with another cell, neuronal or otherwise, through electrical or chemical processes. There are almost 40,000 new synapses are formed every second late in the 3rd trimester. Again, the period of greats of synaptogenesis. And I don't know about all of you, but what I would give for 40,000 new synapses every second, that would really be a life changer. Right, so there's the amazing brain development going on in utero. But another reason that we want to bring this FOAD approach to ASD is that it's relevant because of how the early onset of this disorder. And just picking 2 relatively recent findings, we know about Avi Klim's work being able to identify a decline in eye fixation between 2 to 6 months that seems to be a risk factor for autism. And some of the other work indicating abnormal brain connectivity prior to symptoms as early as 6 months. So clearly this is a disorder that is happening early and really suggests that the contributions, the shaping of it is happening in utero. Also, prenatal morbidities are a significant risk for autism and therefore make FOAD, we should be looking at prenatally and make FOAD relevant for ASD. So specific prenatal morbidities, they're thought of as proxy variables for a compromised in utero experience, and they themselves are associated with risk for ASD, such as abnormal presentation, umbilical cord blood complications, fetal distress, birth injury or trauma, multiple birth, low birth weight, small for gestational age, congenital malformation, and also a deviance in the growth, the fetal growth at either extreme. Too much and too fast or too little also are associated with risk for autism. And then there are maternal morbidities during pregnancy that are a risk for autism. Obesity and gestational diabetes, preeclampsia, and the particularly infection, illness, and immune activation, which is probably underlying a lot of those different morbidities. These are all happening, can be happening in pregnancy. They are a risk in themselves for autism, and again, supporting this idea, we need to bring the FOAD model into the work with autism. And then finally, there are various maternal experiences, which again are exposures for the fetus, which are risk factors for autism. And I'm going to be focusing on 2, and as you've already heard, they're relevant for the work that we do in my lab. So one of them is maternal stress, has been linked to a risk for autism, and the other is SSRI, serotonin reuptake inhibitor, the psychiatric medication. Taking it during pregnancy is a risk for autism. So we put these together, all these different factors happening in pregnancy, the underscoring of how much brain development is going on in pregnancy, and we can really, I think, see why a FOAD model is so important to bring into, again, the world of autism research. But what's the common ground here? What is going on? I would argue that why these factors are all risks for developing autism is because the fetal brain is being affected by each of them. So if the fetal brain is being affected, its development is being affected, and we want to see that, and we want to see it in order to understand the roots of autism, what we need to be able to do is see if we can assess fetal development. So that brings us to the question of how do we ask the fetus questions? That's a little bit of a challenge, but we can do fetal neurobehavioral assessment. And before I start showing you some of the probes that we do to ask the fetus questions, I wanted to show you an ultrasound tape of a 3rd trimester fetus and have you, oops, I beg your pardon. What happened to my film? Did you all see it there? I saw it there. Yeah. Oh, there it is. I beg your pardon. Okay, let's try this again. No. When I press it here.
A little higher on it.
Yeah.
Higher, yeah.
That's a challenge to my glasses. Okay. It's the fetal heart rate, very fast, 120 to 160 beats a minute. And you can see the body here. This is the head. It's a cross section of the arm. And the ultrasound is right on the heart. You're going to see one in a minute without, now you're going to see a little bit better of this baby. Another baby, actually. Here you go. This is the mouth. You can still see the heart beating. This is the head. So it's a parallel view. You can a little bit see the nose right there for a minute and still see the heart beating. These are the legs. There's a good image of the heart. There's a cross-section of the arm, and you just saw another, there you, good image of the brain and the heart moving. So those are it's one image of our research subject. I'm gonna give you another picture of a research subject in a minute. But that is our subject, and now I want to discuss how do we ask the fetus questions, and I'm going to give you an example of 2 indices we use for the assessment of fetal behavior and development. We can look at heart rate and heart rate reactivity, which can tell us about the development of the autonomic nervous system. And we can also do something which is looking at the cross-correlation between heart rate and movement changes and how well they're integrated. We call this coupling. I'm going to use that term a couple times, coupling. And that's letting us know how the autonomic nervous system and the somatic systems are getting coupled, getting integrated, which is a representation of, and a reflection, excuse me, of central nervous system development. I'm later going to show you data that is similar to a growth curve chart on a baby, on a child. We know that coupling goes up reliably over gestation, again, representing the central nervous system developing. So to ask the fetus these questions, we use a Toy2 Doppler actocardiograph for detecting movement and heart rate. And what you're seeing here is a raw waveform of the heart rate. And then here is the movement, and this is in arbitrary units. And what I've circled here is a moment of coincidence of change in both of those signals that would contribute to the calculation of coupling. Okay, so to look at maternal experiences, these fetal exposures that are relevant to autism, I'm going to start with the one I mentioned first, which is stress, maternal stress, and particularly I'm going to look at acute stress. Because if stress is affecting fetal development in ways that has implications for autism risk, we should be able to see the communication of maternal stress to the fetus as an acute transmission. And that's what we're going to try to look at. But it's often humbling in research. You realize that people have asked the question before you got there as well. So in fact, there was a study back in 1967 with a sample of 2 pregnant women. And the question was, can we stress the woman and get the fetus to show us a response? And this is how they stressed the woman. They told her she was breathing a gas which contained only half the amount of oxygen necessary to support fetal life, but that her normal body mechanisms would probably compensate for the altered environmental condition. So, yeah, exactly. All right, so we do not do that. Am I still loud enough for you all? You doing okay? Okay, because I know I move around a lot. So needless to say, this is not how we stress our women, but it was very effective. So this is showing you, and they call it a psychic stimulation. So this is showing you the fetal heart rate is on the bottom here, and here's the maternal heart rate. And you can see that they are both going up together as the introduction of this very threatening statement to this unfortunate pregnant woman. So we do elicit stress in the lab, but we do it in a kinder and gentler way. We give a challenge, a Stroop color word matching task, which if many of you remember your Psych 101. It's a standard cognitive challenge. I'll give you a little bit more about it. We give them a breathing task, which we call RESPAR, so they have to pace their breathing. Each of them is 5 minutes interspersed with a rest period. So for Stroop, you need to, the correct answer is the color of the word, not the word itself. And it goes very fast, and you're told when you're getting it wrong, and it's frustrating. This is our breathing task where we force them to, we don't force them, we ask them, and they are rather compliant to breathe at the rate of this bar. And we also then, that's a pretty typical breathing rate. And then we ask them to breathe at this rate as well, which is quite fast. And if anyone is taking yoga and is used to being told how to breathe, that in and of itself can be rather frustrating, particularly for control-oriented people like us New Yorkers. And when you're pregnant, as you may know yourself or from people in your life, particularly in third trimester, which is going to be the major participants in our research, or in the third trimester, your breathing is already going very fast. So it's quite frustrating to be told how to breathe. So this is another view of our research subjects, and yet another. So I'm first going to show you what happens to women's cardiorespiratory activity when we give them these tasks. And what you're seeing is diastolic blood pressure, heart rate, and systolic blood pressure, and then respiration rate. And so what I've circled here is the significant changes in systolic blood pressure and respiration that are happening in that Stroop period. Now, interestingly, we don't get any significant changes in the breathing task, this RESPAR task, the fourth one here. We are eliciting very big changes in their breathing, but from fast to slow in these pregnant women, and that probably equalizes it out the way that we've analyzed it here. But really the take-home message from this slide is that women are reacting to the Stroop, and they're not really reacting, certainly not in other indices besides respiration, to the RESPAR task, the breathing task. So what does the fetus do when we've gotten the mother jazzed up this way through our paradigm? Well, interestingly, and this is work by Hannah Gustafson, who's a postdoc in my lab, the fetus is not showing a heart rate change to the Stroop task, but by the time we get out to this paced breathing, the fetus has a significant heart rate change. So what we're interpreting from this is that there's the fetal heart rate response is not merely in line with the maternal stress-induced biological reactivity, but we do see the fetal heart rate response to a sequence of maternal challenges indicates the maternal experience is getting in. It is affecting the fetus. So now I want to move on to look at, chronic distress. And I'm using the word distress purposefully as an umbrella term for depression, anxiety, and stress, which tend to be highly correlated. And the data indicating that stress is associated with risk for autism comes in large part from the ice storm studies up in Canada. So that was a more chronic experience than just an acute 5 minutes in the lab. So can we see chronic stress being associated with any differences in the fetus? So I'm going to walk you through this very complicated slide, but on the bottom you should now be familiar with our paradigm of having two challenge, the Stroop and the paced breathing, and this is fetal heart rate, actual fetal heart rate here. And if you look at the green line first, you're seeing a replication of what I already showed you. So these are fetuses of women who are healthy controls. They've been screened. They do not have a psychiatric illness, the mothers, that is. Fetuses show no reactivity to Stroop, but by that paced breathing, they do. What I would like you to focus on is the reaction to Stroop. These are fetuses of women in the red who have comorbid depression and anxiety by a psychiatric interview, and they also have, or they just have, their mothers have a major depression. They are showing a statistically significant heart rate change to the stressor task that the mother is going through. Now, very importantly, by group, the mothers are not showing any differences in their cardiorespiratory reactivity. So what it would be like is that if someone ran in the room right now, some of us would jump, and some of us would be much slower to respond. So the comparable stimuli is coming into the fetus, but their fetuses are differentiating themselves based on the chronic exposure that we think they've had from their mother's distress, identified in the psychiatric illness. So this slide is really a summary of what I've said, and we think that this acute stress activation during the Stroop task, like the door, is a stimulus for some fetus and not for others. And it's by the third trimester, the fetuses of psychiatrically ill women, those chronically distressed, are more reactive. And to use Barker's words from one of my first slides, perhaps these fetuses have adapted. They've been programmed by third trimester by this altered environment associated with their mother's psychiatric illness, so they respond differently to comparable maternal physiologic activation. So I want to move on and look at chronic distress in relation to fetal development over gestation. That was just a one-time point in third trimester. And to do that, I'm going to call on Janet DiPietro's work, and this is where I'm going to show you those what look like growth curve charts of what fetuses do over gestation in a very reliable way. So these are data from 2 samples of feti, one in Baltimore, Maryland, and one in Lima, Peru, and what their behaviors are in heart rate and coupling over gestation. So you can see over gestation, on average, the heart rate is going down in these 2 groups of feti, and that heart rate movement coupling is going up. But I'm sure you're also noting some differences in magnitude and also some differences in the shape, the trajectory of those changes over gestation. So what we can take from this is that there's consistency in developmental patterns, again, like a growth curve chart for a child getting taller each year. But there are also geographic differences, and the fact that there are geographic differences lets us have the idea that perhaps there are maternal experience differences as well that may be associated with fetuses having differences in their development over gestation. So we took on this question and based this work over the course of pregnancy on ambulatory assessment of mood. So over 24 hours, women were using these Palm Pilots to mark off their moods. And in hindsight, I kind of think it was torture. They had to answer every half hour. I think it was 17 questions of how much you're feeling happy, sad, joyful, angry. It was, I would never now do this to anybody. It's really tedious, but we got a lot of great data. And we also were collecting cortisol on a diurnal, getting a diurnal slope of cortisol over actually 48 hours. So what did we find? We found greater distress early in pregnancy was associated with a lower heart rate over gestation, and this was especially true in males. And this is work that Colleen Doyle, who is in my lab and is now at University of Minnesota, took the lead on. And we also found that greater cortisol early in pregnancy was associated with a flatter slope and a higher level of coupling over gestation. So you can see the slope is flatter, but they are starting at an earlier point of being higher up in that coupling index. So we're seeing maternal chronic distress in fetal development is associated with lower heart rate. That's the expected trajectory of development sooner. They're getting there sooner. And we also are seeing, associated with maternal distress, that over gestation, this fetal development has an increase in coupling with an indication of starting higher. So the expected trajectory of developing sooner. So they're at a higher level of their coupling sooner, and their slope is flat because they've already gotten there. So the last exposure I want to focus on in terms of asking the fetus questions, and one that has relevance for understanding ASD because it's been linked to ASD, is SSRI use in pregnancy. And this is work that, again, Hannah Gustafson was involved with, and it's from a data set of Zach Stowe and Jeff Newport, who previously were at Emory. So this is a really fantastic data set of 161 pregnant women, all with a history of major depressive disorder. And that is key, because so many, as you probably know, so many of the studies that are looking at effects of SSRI use in pregnancy on child outcome across many areas, but certainly across looking at autism as an outcome, have a confound of indication. So the women who are taking the SSRI are often much more sick, and they're being compared to women who don't share a psychiatric history, who are, if you will, squeaky clean controls. So in that circumstance, you really can't tease apart whether it's the severity of the illness or the history of having the illness from the actual medication. And here we have a very homogeneous sample, all of whom share this history of major depressive disorder. And we're going to do our usual paradigm of asking our fetuses questions, but a shorter version of a baseline Stroop and then the recovery. And I should say that the depression was very well controlled in this group. 70% of the women were taking psychiatric medication, and of those, 55% were on an SSRI. And we grouped the medications into no medication, SSRI only, SSRI and other only. So I first want to show you, what did the fetuses do over gestation? And here you're looking at, it is true, there's data on our paradigm, but for just a minute to concentrate on, this is 24 weeks, these fetuses. This is their average heart rate, and then by the third trimester at 36 weeks, their average heart rate. So what we're seeing is across all these subjects, the fetuses are doing what's expected. They are decreasing their heart rate over gestation. And we also saw that across all fetuses, there were almost no effects of medication. The one result we had was that SSRI use was associated with overall more fetal movement. Now, in terms of what did these fetuses do in relation to eliciting that acute stress from their mothers? Well, at 24 weeks, maternal distress was associated with an increase in fetal heart rate during Stroop, just as we'd seen before in data from my lab, 36 to 38 weeks. Now, interestingly, no result at the 30-week time point, but when you go out to 36 weeks, distress was associated with a decrease in fetal heart rate during the Stroop task. And we often think of a decrease in heart rate as an orienting response. When we recognize something, we're turning our attention, we decrease our heart rate. And we wanted to probe this a bit more, so we pulled out the 98 of the 161 fetuses who had data at both time points. 46% of those showed this fetal heart rate increase during the first Stroop exposure during the first session. And of those, 67% had a decrease in heart rate at the third testing session. And I'll come back to that point. So looking at fetal behavior in the context of controlled depression and medication, we see that the SSRI use was associated with more motor movement, really no findings for other medications, and modest findings for maternal distress in a sample with very low distress or depression. So we saw fetal heart rate reactivity changes from an increase at time 1 to a decrease at time 3. And we think this may be possibly consistent with the idea of priming. And priming is a form of an implicit memory in which exposure to a stimulus at one time influences the response to the other. So that's a hypothesis we're making on our own results and something, and it's an interpretation and now a hypothesis we want to further test. So I asked early on, can we assess fetal development, and in a way that's going to bring FOAD into ASD, to use these acronyms, that can help us understand ASD. We can ask the fetus questions. We can ask about fetal neurobehavioral development, and I believe that these results are relevant to our pursuing understanding of ASD. And the results are relevant in particular because the maternal experiences that I've been describing as related to these fetal behaviors are implicated in ASD risk. And as I said, they're also associated with characteristics of fetal development. So we saw maternal acute stress being transmitted to the fetus, and we saw maternal chronic distress and cortisol, of course, which is an indicator of a stress activity, being associated with greater heart rate change during maternal challenge, a change in heart rate during maternal challenge possibly priming by an earlier exposure to the same challenge, and lower heart rate and higher coupling level over gestation. And then we saw maternal SSRI use was associated with more overall movement. So we really can ask the fetus questions. But one of the main questions about asking the fetus questions is, does the fetus tell us about the child? So here I want to use, again, this idea of multi-level analysis, looking at trying to make the connection between how the fetus is and how they are as a child in two different ways, looking at infants and looking at infant brain. So first, this is work from my very close, oh my goodness, I cut off her name, that's terrible, sorry, close colleague, and Elizabeth Werner. We've been working together for a long time. And what she was asking in this study is of these fetuses who showed a significant heart rate response during mother's stress versus the fetuses who did not have a response, no action, didn't show that they cared at all, what are they like as 4-month-old infants responding to novelty? And this is a very standard paradigm to do with 4-month-old infants. And perhaps, I guess you can read in the yellow there, that fetuses who were reactive went on to be much more during mother's exposure to stress and that Stroop task, went on to be much more reactive, particularly motorically, as 4-month-old infants. And I'm going to show you 4 clips, and I hope you will all score well and agree with, you haven't learned the scoring pattern, but let's see what you think. After a while, if you feel like it, you can shout out lower high motor, lower high reactive, but maybe you want to get some comparisons first. I'll give you some comparison. We can also just watch babies all afternoon, and that'll call it a day. So try to think about what you think they were like as fetuses. Okay, 2 more. Hold your votes, unless you get an idea. Okay, and our final one. Okay, any thoughts? It's a little subtle, but not when we do the coding. The first 2, low reactive, and the latter 2 are high reactive. You should see what she starts doing with her poor bow after a while. All right, so the same stimuli. So we're able to make this connection between what are the babies like as fetuses and then what do they go on to be like at 4-month-old. So the fetuses, as indicated, that with a heart rate increase to acute maternal stress, they're more likely to be behaviorally reactive to novel stimuli at 4 months. And you could sort of see in the first more high reactive baby a hint that he looked more focused and attentive. So that's more of an observation. It's not part of our coding scheme. All right, I want to move on to our work using functional magnetic resonance imaging, and this is part of a project headed by Jay Gingrich and John Posner and Chai-Kuk Cha took the lead on this. And here we're going to be looking at maternal depression and the infant's amygdala prefrontal circuit. And so I'm going to really, these are sleeping infants. And I'm going to jump to one of the primary findings we have so far is that infants exposed to prenatal maternal depression, we're going to call it PMD, you'll see it in the images, show more inverse functional connectivity between the prefrontal cortex, largely in the dorsal area, in relation to the amygdala. So that inverse activity, the more the prefrontal cortex is working, the less activity in the amygdala. So really just showing you the areas of the brain that have been identified in this project, and then showing you the magnitude of the differences. So here you see the PMD, these are about 4-week-old babies, have greater inverse connectivity, again in the prefrontal cortex and the amygdala, compared to these fetuses of healthy control women. Now this was very intriguing to us following on Nim Tottenham's work, and I'm going to show you some results of hers and walk you through it and make the connection. In her work, children with early adversity, which was being in orphanages for the first 2 years of their lives, show more inverse, and they called it mature, amygdala medial prefrontal cortex connectivity. Now this is doing a paradigm, a fear paradigm. But let me walk you through the results. What you're seeing here is the comparison group. For the children, they do not show this inverse activity in terms of the prefrontal cortex and the amygdala, but by adolescence they do. And so her interpretation is that as children, you have your parents to help you with this affect regulation. But interestingly, the previously institutionalized children, those who had been in orphanages the first 2 years of their lives, show this inverse connectivity, functional connectivity, earlier than the typically growing children in their homes. So they interpreted this as a mature connectivity, and we are relating being in utero with a mother who has depression as a kind of adversity, and seeing an inverse connectivity and interpreting our finding as also possible maturity. So infants, we have one more finding with this, is that infants' amygdala prefrontal connectivity is also related to our fetal heart rate paradigm. So again, these fetuses who have this reactivity during the paradigm in the red and the purple, they tend to have more of this inverse connectivity. So here's the amount of the inverse connectivity, and then on the y-axis is the fetal heart rate. So they are traveling together, and at a minimum, their indications we're now interpreting as more mature development. Having this more reactive heart rate at this time is in that it goes with this functional connectivity that seems to be online before its time, based on Nim's interpretations. So in our fetal to infant studies, if we take it together, and I've already given you a spoiler of what our interpretation is going to be, but we're seeing that the data is suggesting maternal distress is associated with accelerated development. In the fetus, we're seeing more reactivity to maternal challenge. That change in heart rate during maternal challenge may be priming. So the fetus has a heart rate reactivity later on. We saw it in the data from the Emory group with whom we were collaborating. And we saw lower heart rate and higher coupling over gestation. And in the infant, we saw more reactivity in their behavior to novelty. Possibly, clinically, it feels like more aware of their environment. And we saw this greater inverse functional connectivity between the amygdala and the prefrontal cortex. So accelerated development is a very elegant interpretation that is used a lot in FOAD research. And the idea is that prenatal distress from the mother is an exposure for the fetus that is forecasting an adverse environment. So it has an evolutionary function. It's know your enemies. It's going to be a hostile world out there. And the fetus responds with accelerated maturation. You could think about this as adaptation. So the fetus starts, as we learned in Barker's terminology. The fetus spends less time in a non-optimal environment, and the mothers limit their energy cost of the individual pregnancy in the face of a possible hardship. But we can also wonder, is this accelerated development, or do we want to use a somewhat different perspective to understand this, and that is thinking about critical periods. And I know you just all had Takao Hench come and speak here, and I'm referencing his work with this idea. So critical periods, they're window during which a system is open to structuring or restructuring on the basis of input from the environment. And input is necessary to trigger the opening of critical periods. But what processes open critical periods, mediate them, close, or reopen them is very much of interest and related to exposures that may happen in utero that affect fetal development. So here are some images from this recent paper that he and a colleague did illustrating this idea of critical period. And you can see a typical critical period, but you can see a precocious critical period or a delayed response, an extended critical period, one that stayed open, never closed. And in largely animal work and then some human studies, he's looked at different, what he calls experimental manipulations. I'm using the term exposure because we can't experimentally manipulate our women. He's seeing these exposures, these manipulations related to shifts in these critical periods. And in particular, in utero depression exposure in his hands seem to be extending the critical period. And we can wonder if that may map onto our results around accelerated development. He also was seeing that increased arousal, and we can wonder if maternal stress is, as we saw the fetus is reacting to it, he finds that that extends critical period plasticity. So we can think about that there's this effect on fetal development, but maybe what we're doing is really altering these critical periods. And in altering critical periods by these exposures in utero, we're altering what inputs get in and for how long they have an influence and to what extent the fetus adapts to them. So this seems to me a very good frame for beginning to think about what's going on in the prenatal period, bringing FOAD to ASD and thinking about these exposures and then having the tools to track how this might relate to risk for autism. So really I've gone through so far, and I just have a little bit more to go talking about transmission from mother to fetus. But I've touched on these different prenatal experiences to make the point that FOAD is relevant to ASD. And I really focused, of course, on these maternal experiences, the fetal exposures. And I've wanted to suggest that the fetus and the infant, their development is being affected in utero in ways that has relevance for ASD. And then, again, that maybe what's really happening is these critical periods are being affected. And we can look at that by the fetal tools that I've described and by infant assessment. And we can study these processes and see how they're relevant to the risk for autism. I'm wanting to touch on one more topic before I wind up and have some thoughts about where we could go next. One of the major questions is, how does maternal experience affect fetal behavior and development? And again, taking a multi-level approach, having an idea and then showing you some data. Sensory processing and genetics are two pathways. And to do this, I'm going to first go back to an interesting article. I don't know if any of you saw this in the Times last summer. It was a very interesting piece, right? So for those of you who didn't, there were two pairs of identical twins who were raised as two pairs of fraternal twins. And then they actually found each other. It's a remarkable story. But I have to say, personally, what was remarkable to me is what the journalist highlighted. And she said that even the identical twins are already different products of their environment being whatever intrauterine conditions rendered them separate in the first place. And she's right on target. Even identical twins come out weighing different weights. Something is different in the environment for them. But she went a step further, and she said these environments are different, for example, perhaps because one twin is further from the sound of the mother's heart, its reassuring steady beat, sending that child on a slightly different life course. So that gets us to think about a stimulating environment and how women's lives, of course, their individual differences and what that means for fetal development, and perhaps that a stressful life that a woman is having is getting to the fetus. I've already showed you some evidence for that, and we particularly saw it in the acute stress transmission to the fetus. So through all the body changes and physiologic changes, the heart rate, the blood pressure changes that are stimuli for the fetus, their sound for the fetus, their vestibular changes, they're arousing for the fetus. They can amount to different stimulating environments from one woman versus another. They're going to be part of contributing to that fetus's environment and how that fetus adapts to the environment and what that means for their development. Another path for communication of maternal experience to the fetus is what we might call a direct transmission. And here you have the psychological experience of distress, which is, of course, a biological experience as well. And I'm just picking here 3 of the primary potential pathways by which maternal experience may get to the fetus. And I'm really just going to touch for now on cortisol. And the thinking is that the elevations in cortisol, for example, in association with feeling very stressed, is going to mean an altered environment for the fetus in ways that could affect central nervous system development and ultimately the child's trajectory. So very briefly, the HPA axis in pregnancy. As I've already indicated, and many of you already know, it's a major effector of the stress response system, and there's some data that's elevated during depression. And prenatal cortisol, I'm just picking a few of the results. It's associated with hyperactivity in the amygdala, so you can store that in the back of your mind in relation to our prefrontal cortex amygdala findings. And it's also associated, for example, with variation in neuronal migration. The pregnancy is a hypercortisolemic state, and I'm going to show you a very complicated image, but we're going to break it down. And in particular, what I want to focus on in this image is an enzyme called 11-beta-HSD2 that is very strongly expressed in the placenta, and it inactivates cortisol to cortisone. So what the image you're seeing here is the typical HPA axis that we all have, with its very well-functioning negative feedback loop. And then, of course, the fetus is developing its own HPA axis. But here what you have is in the placenta, this enzyme, which again is inactivating cortisol once it crosses the placenta to be in the fetal compartment. And one thing that's different about how the placenta functions as an endocrine organ is that it is responsive to cortisol in a positive way. So it pumps out more CRH in response to cortisol. So through maternal distress, you can have some significant positive feedback loops just looking with the mother and the placenta to have a very elevated state of cortisol secretion. And so what I've given you right now suggests that we need to, it's going to be complicated to think about how is cortisol getting to the fetus and then potentially having effects on the fetal brain. And I'm going to show you data looking at the association between cortisol in maternal plasma and cortisol in amniotic fluid that suggests just how complicated it can be. These are results from Vivek Glover. And what you're seeing here in the box I've just highlighted is for women who are low anxious. There's no correlation, a 0.05 correlation between cortisol in the amniotic fluid and cortisol in her plasma. But if you travel down these panels to a woman who's high anxious, there's a very strong correlation, suggesting that something about being anxious, being self-identified as high anxious, that goes in that distress category, is having an effect on the placenta so that the 11-beta-HSD2 is not functioning as well in the fetus. There's more cortisol in the amniotic fluid and the fetus is getting exposed to more of it. So this really brings us to question gene expression in the placenta. So very briefly, when we're talking about gene expression, we're talking about changes in gene activity that don't affect the basic structure. And we're going to be, particularly in the data I'm going to show you, be looking at DNA methylation. And so DNA methylation, of course, is turning on or off the expression of the gene. And we're going to particularly be looking at methyl marks. So this is results that actually were just published last week with my colleagues Francis Champagne and Ben Tycho. And I'm going to next show you a very complicated data and walk you again through it. What we are looking at is methylation in 11-beta-HSD2 that produces that enzyme that protects the fetus from active cortisol in relation to maternal stress, perceived stress, PSS, and then also in relation to that fetal variable I told you about, fetal coupling. So what you have here on the left is PSS, the perceived stress in the woman on the y-axis, and methylation down here. And what you can see is that the lower the stress, that was associated with lower methylation. So that would mean that the fetus is being better protected from that. There's the expression of that gene, and the enzyme is working, protecting the fetus from more cortisol. And you see indication, and statistically true, that the higher the stress, the higher the methylation. So that gene is getting inactivated. We'll go over here, and we're going to be looking at that gene, methylation of that gene in relation to fetal coupling. And what you see here is if we have low methylation, again, that gene is functioning well to make cortisol inactive, is the thinking. We don't have proof of that piece of it. But less methylation of that gene, so the gene is getting to function, we have a higher level of coupling. And remember, we expect coupling to increase over pregnancy. And then you can see when we have high methylation, that gene is not being allowed to do its job, we have lower coupling. And just as one note here, we have the two red triangles are fetuses of donor eggs, pregnancies. So we're very interested in moving in that direction where we can begin to tease apart some shared genes and really isolate the in utero environment in relation to maternal experience. So this worked out as a mediation model for us statistically. So we can go from the level of perceived stress in the second trimester to having more methylation of that gene, so that gene is not able to do its job and be expressed so the enzyme can protect the fetus from cortisol, and that's associated with lower levels of fetal coupling. So here I've wanted to show you some data for how it might be that maternal experience is transmitted to the fetus and affects fetal development. And I want to really wind up the talk now of asking in just a few slides, what can we do now? And that really is, what are our next steps potentially to expand prenatal studies focused on autism spectrum disorder research? And I'm going to describe a couple of projects that are emerging and I think very promising. One of them is looking for prenatal risk markers for ASD using two different samples at risk. And one of them is, and I should say with colleagues Bill Pfeiffer and Michael Myers, and this is Jeremy, whom I already mentioned. One is to look at babies who are born preterm because they are both at risk for autism, and looking at a particular activity, electrocortical activity, and seeing if we can tease apart better what is going on that puts these babies at risk. And the other is to look at blood serotonin levels and really look at both fetal, newborn, and infant brain and behavior. And as you all probably know, elevated blood serotonin levels is one of the most heritable biomarkers associated with ASD. So this is a collaboration going on with many different groups at Columbia, including this CTNI initiative in color here. I think we also really could be leveraging the OB tools, meaning obstetricians, and we could be collaborating with them, and we can be doing some interdisciplinary work. They are already studying samples at risk for ASD. As I already indicated, women with preeclampsia or fetal growth, they have fetal growth restriction, and they're using their assessment paradigms to predict compromised neurodevelopmental outcomes, such as mid-cerebral artery blood flow. And here's just a cutout of an article representing that kind of work. So we can really partner with them to have different disciplines working in this area. And then we can use existing prenatal data sets. I was speaking earlier with Julie about this. There's a request for applications from the NIH called ECHO, Environmental Influences on Child Health Outcomes, wanting that nationwide many cohorts will be brought together. That's one of the strengths, but in work with which I'm involved, many people are involved it's wanting to build on the fetal growth study. These were nearly 3,000 women nationally represented by race and ethnicity who had serial 2D and 3D ultrasounds. You can look at cerebral volume, head circumference, and now the children are 4 years plus. And just being very conservative, 34 of them will have really frank ASD, autism, I should say AD. And of course, that's a very small number, but there's phenomenal prenatal data to start generating some hypotheses. And then finally, in conjunction with Don Pfaff and Daniel Keenan, and Donald, I think, is very known to this group, we have more precise ways of acquiring maternal heart rate and fetal heart rate actually using EKG methodology. And with that, we can be looking at the fetal heart rate with much more precision and the maternal heart rate. And Dan is very interested in modeling that using mutual information modeling between these two systems. And here's just a representation, which I'm not going to go into, but really the promissory note is, can we look at maternal-fetal communication in utero? Can we really develop a tool for doing that? So what I've wanted to suggest today is that FOAD is really relevant to ASD, and we can be putting, if you will, adding this fetal assessment approach that is informed by FOAD into this wonderful multi-level model that of course, Jeremy put together. So we can really augment this multi-level analysis of ASD and ultimately to improve health outcomes via earlier detection, targeted intervention, better knowledge of the complex etiology. And we really have the tools to start studying early development in multiple ways. So with that, I'm going to stop and thank funders and thank all of you and collaborators, and really welcome any questions and the discussion. Thank you.